I might not even read my title and just start. But yeah, thank you to the organizers. I'm enjoying the conference and I look forward to the remaining days. So the development of a multicellular organism begins with a period of intense cell cycle activity. In the early Drosophila embryo, nuclei undergo a series of 13 rapid synchronous nuclear divisions. And during this time, patterns of gene expression form across the axes of the embryo. For example, the zygotic gene hunchback is expressed primarily in a broad domain in the anterior of the embryo. So how do these gene expression patterns form? They're largely controlled by the activity of transcription factors, which bind to DNA sequences that are responsible for regulating target gene expression. And in many developmental contexts, transcription factor gradients define spatial domains of gene expression. So some targets are only expressed across high transcription factor concentrations, while others are expressed across a broad range of concentrations. Different molecular mechanisms have been proposed for concentration-dependent transcriptional regulation. Target enhancers can have differential affinities for transcription factors. Additionally, enhancers can show different degrees of cooperative transcription factor binding. And the early Drosophila embryo is an excellent system for studying concentration-dependent transcriptional regulation because the embryo expresses a precise exponential gradient of the transcriptional activator Bicoid across the anterior-posterior axis, with the highest Bicoid concentrations in the anterior of the embryo. Bicoid binds to its target enhancers in order to upregulate precise domains of gene expression across the AP axis. The posterior boundaries of these gene expression domains align with different Bicoid concentration thresholds. However, we can't predict from the Bicoid binding site number or affinity within enhancers where the enhancers will upregulate transcription along the gradient. So it remains an open question. What is the molecular mechanism of Bicoid concentration dependent transcriptional activation? I've been using the Hunchback P2 enhancer as a case study for understanding concentration-sensitive transcriptional regulation. Hunchback P2 is a 250 base pair enhancer, which is responsible for mediating an anterior domain of hunchback expression. And we know that this enhancer is very sensitive to Bicoid concentration because as you increase the magnitude of the Bicoid gradient in the embryo, the posterior boundary of this expression domains shifts towards the posterior. So using this enhancer as a case study, I've been working to model the molecular mechanism of concentration-dependent transcriptional activation. I first sought to determine an informative output to model. So I found a transcriptional signature of Bicoid concentration. I then built a model to test hypotheses about how aspects of the genomic environment and early development, such as chromatin and the cell cycle, influence concentration sensitivity. In order to assess transcriptional outputs, I created a reporter for the Hunchback P2 enhancer. This reporter allows for live measurements of transcription using the MS2 MCP system. So in this movie, the yellow spots are points of nascent transcription, and I've colored each of the nuclei based off of the relative intensity of their transcriptional foci. You can see during mitosis, there is no transcription, and then there is a delay followed by heterogeneous onset of gene expression across the domain. And I quantified the times following mitosis that each of the nuclei initiate transcription, and then plotted these transcriptional onset times against the AP positions of each nucleus. And I found that the mean and variance of the transcriptional onset times correlate with AP position. So as Bicoid concentrations decrease along the AP axis, onset times increase. This correlation suggests that Bicoid concentration regulates onset time. But I wanted to test this experimentally. to test whether Bicoid concentrations determine the timing of transcriptional onset. So I created mutant embryos that instead of expressing Bicoid in a gradient, they express Bicoid uniformly across the embryo. And in these embryos, the hunchback P2 MS2 reporter is also uniformly expressed. I quantified the fluorescence intensity of GFP Bicoid in the graded and uniform lines and found that the uniform line expresses Bicoid at the concentration normally observed at around 37% egg length in wild-type embryos. I then quantified transcriptional onset times in both the graded and uniform lines, and this quantification shows that the onset times distribution to overlap around 37% egg length. So this experiment demonstrates that Bicoid concentrations are very precisely determining the mean invariance of transcriptional onset times. So onset times are informative about the regulatory role of Bicoid concentrations. How can I model them? In building a model, it's important to consider that transcriptional activation is stochastic. So at a given position across the AP axis, all the nuclei don't initiate transcription simultaneously, but there are distributions of onset times across the AP axis. So predicting transcriptional activation requires a stochastic modeling approach. I built a two-state model of promoter activity where following mitosis, a nucleus begins the nuclear cycle in a transcriptionally inactive off state, and then the nucleus can transition into an on state with an average rate of K on. And transcriptional activation is a random process. So as a starting point, I propose that transcription spots appear according to a Poisson process. An example of one of these processes would be if you're standing by the street watching cars go by, every time, like cars pass you after random time intervals. So I thought, okay, I'm live imaging an embryo, watching spots appear. An event is when a spot appears. This could be a Poisson process. So I asked, how long would I have to wait before I see a spot? In the case of a Poisson process with a constant rate lambda, the waiting time until one event is described by an exponential distribution with a mean of 1 over lambda. However, the onset time distributions are not exponential. So Poisson process with a constant rate does not describe spot appearance. Why could this be? This is because in early development, genes are transcribed during this period of intense cell cycle activity where there are very frequent mitoses and nuclear cycles are very short. And so there's a significant variation of nuclear transcription factor concentrations over the cell cycle. Additionally, transcription must be coordinated with genome replication during this time. Therefore, to model hunchback P2 MS2 activation, you must know the Bicoid concentrations over space and time. To estimate Bicoid concentration, I applied a model for the Bicoid gradient, which takes into consideration the synthesis, diffusion, and degradation of the protein. In order to get a sense of the dynamics, I live imaged GFP Bicoid across nuclear cycle 13. I then normalized the fluorescence measurements in each of the nuclei, finding that they all follow pretty similar dynamics, normalized dynamics. And then I averaged these dynamics. And I multiplied the prediction from the gradient model with the normalized dynamics at each time, normalized fluorescence levels at each time point to get a sense of how Bicoid concentrations in nanomoles change over space and time. Therefore, I was able to incorporate these Bicoid concentrations measurements into the expression for K on. And then I asked, okay, if K on varies according to Bicoid concentration, can the model simulate onset times? So I applied this, the following stochastic simulation algorithm. So for each nucleus across the AP axis, the algorithm calculates the nuclear Bicoid concentration at that position and time point, then estimates K on from the Bicoid concentration, then draws a waiting time for the off-on transition from an exponential distribution for a process with a rate parameter of 2 times k on, because there are 2 copies of the reporter in each nucleus. Then if the waiting time is sufficiently fast, the algorithm records the onset time of the nucleus as that time, but if it's too slow, then it proceeds to the next time step. So I'll go through a quick example. Say you're simulating a nucleus at 35% egg length, and you're simulating with 1 minute time steps, which are very long time steps, but just for the graphs, I'm saying that you're simulating with 1 minute time steps. So at the beginning of the nuclear cycle, Bicoid concentrations are low. With the Bicoid concentration, you estimate k on. You then draw from the waiting time distribution associated with k on. And if it is longer than the time step, then you proceed to the next time point where Bicoid concentrations have increased. You then re-estimate k on. Now that your waiting times have shifted towards smaller time intervals, and you're more likely to draw a smaller time and record the onset time. So this simulation approach relies on understanding how k on depends on Bicoid concentration. So what is an appropriate equation for k on? With the model, I tested the hypothesis that Bicoid could be interacting with open chromatin in order to upregulate transcription. And one way you could reflect Bicoid's interaction with open chromatin is simply to linearly relate k on to the normalized Bicoid concentration. However, with this definition, while the model can simulate an increase in the mean invariance of onset time across the AP axis, it doesn't simulate the posterior boundary of expression. I then tried a nonlinear relationship of k on to Bicoid concentration by defining k on according to Michaelis-Menten reaction kinetics. In this case, the model also can't predict a posterior boundary, and it's also simulating onset times far prior to the 4.5 minute onset time minimum observed in the data. And for both of these models, you can vary the parameters, and no parameter combination allows you to estimate the data. Similarly, I found that if you define k on according to a Hill equation describing cooperative binding, while the model can simulate a posterior boundary of expression, it can't do so accurately with reasonable parameter estimates. And additionally, the mean invariance of the onset times is significantly lower than those observed in vivo. So the failures of these models suggest that Bicoid must navigate additional barriers to bind its sites in the Hunchback P2 enhancer beyond just interacting with open chromatin. So how else can we define k on? We know that Bicoid must navigate nucleosomes in order to bind Hunchback P2. And this was shown by an experiment that measured genome-wide Bicoid binding as well as chromatin accessibility in embryos that express Bicoid at uniform levels, either at low, medium, or high levels. And this experiment showed that as you increase Bicoid concentrations in the embryo, Bicoid occupancy increases at Hunchback P2, and additionally, chromatin accessibility increases as 2 nucleosomes are evicted from the enhancer. So in order to account for this behavior in the model, I applied an existing model published by Len and Mirny in 2010, which is a model for transcription factor nucleosome competition. This is an allosteric framework where a DNA locus can be in a nucleosomal state where a transcription factor binds to its binding sites with a very low affinity, or the locus can be in an open state where a transcription factor binds to its sites with a very high affinity. And I applied this model to the case of Hunchback P2, which contains 2 nucleosomes and 9 Bicoid binding sites. And in order to predict the fractional Bicoid occupancy at all the sites occluded by the distal nucleosome as well as the proximal nucleosome. And then I related k on to the product between these fractional occupancies at the distal and proximal nucleosomes. And with this definition, the model can now simulate a posterior boundary of expression and it can simulate this boundary, predict the boundary accurately, and appropriately predict the onset times at the posterior of the expression domain. So Bicoid-nucleosome competition seems to be setting a concentration threshold for transcription. However, if you look at the onset times in the far anterior of the embryo at high Bicoid concentrations, the model still fails to predict the onset times, predicting onsets prior to the minimum observed in the data and additionally low variance. So what else could be delaying transcriptional activation at high Bicoid concentrations? There's evidence that DNA replication could delay transcription following mitosis, because a few years ago, it was shown that without the completion of DNA replication, transcription is inhibited. Therefore, to model onset times, you must know the timing or be able to estimate the timing of DNA replication at the reporter. But this is something that's very difficult to measure experimentally. So I devised a way to model the timing of replication at the reporter. And I drew upon measurements of the positioning of replication origins within long replicating Drosophila chromosomes. These were measurements made by Blumenthal in 1974. And I'm showing an electron micrograph of Blumenthal where I've annotated each of the origins of replication with a green dot. You see they look pretty randomly distributed along the genome. So if origin spacing is random, then the reporter will replicate at different times between nuclei. So for example, in nucleus A on the left or the right, the origins of replication fire far away on either side of the reporter. And therefore, following the initiation of DNA replication, the reporter takes a long time to complete replication. In an alternative case for nucleus B, the origins of replication fire very close to the reporter. Therefore, the reporter very quickly completes DNA replication following mitosis. So I'm able to estimate the timing of DNA replication by dividing the distance between the nearest the two adjacent origins by the speed of DNA polymerase, which is 5.3 kilobases per minute. And then I can add on the time following mitosis at which replication initiates, which from some experimental measurements is around 4 minutes. So if I want to model on replication times, I must first model inter-origin distances. To model inter-origin distances, I thought of origins appearing along the genome and according to a random process. So I asked, okay, how far must I travel along the genome before I have encountered two origins? For a Poisson process, the wait time, or now in this case distance, until a certain number, or n origins, is described by the gamma distribution, where alpha is the number of events you're waiting for, and beta is the inverse of the rate of the Poisson process. So fortunately, Blumenthal measured the average inter-origin distance, which is 9.7 kb. Therefore, the rate of origin appearance is approximately 2 origins per 9.7 kb. So I defined alpha in a gamma distribution as 2 origins that you're waiting for, and then beta as 9.7 kb per 2 origins. This gamma distribution reflects the Blumenthal's measurements of inter-origin distances. This is evidence that, or it points to the origins being randomly distributed across the genome. And then converted these inter-origin distances into replication times to generate a distribution of possible replication delays for the reporter. So now the stochastic simulation is able to draw a DNA replication delay for each nucleus. So here's an overview of the new simulation approach. So now 2 things must happen for a nucleus to transition into an on state. Bicoid must outcompete nucleosomes at the enhancer, and the promoter of the reporter must complete DNA replication. So there can be a few different scenarios. For nucleus A, Bicoid binds early in the nuclear cycle, but the nucleus can't transition into the on state until DNA replication happens, and therefore it's the timing of DNA replication that determines the timing of transcriptional onset. Alternatively, for nucleus B, the promoter replicates early in the cycle, and the nucleus doesn't switch to the on state until Bicoid binds. So it's Bicoid concentrations that are delaying transcriptional onset and determining transcriptional onset times. So now with the incorporation of DNA replication into the simulation, it now predicts the mean and variance of onset times in the anterior of the embryo where Bicoid concentrations are very high. Furthermore, the model still incorporates nucleosome competition and is able to simulate the posterior boundary of gene expression. And I'm able to annotate each of the onset times based off of whether DNA replication determined the timing of the onset or Bicoid concentration determined onset time. This is just a good way to visualize that the model is supporting the idea that DNA replication delays the onset of transcription at high Bicoid concentrations. So from this study, I'm able to make a few conclusions about the molecular mechanism of concentration-dependent transcriptional activation. In the case of hunchback P2, at high Bicoid concentration, DNA replication limits transcriptional outputs. At intermediate Bicoid concentrations, competition with nucleosomes lead to a posterior boundary of expression. And then finally, at very low Bicoid concentrations, nucleosomes entirely prevent gene expression. And I expect that similar mechanisms are at play at different Bicoid target enhancers, and that the behavior of these enhancers can be modeled given our understanding of the Bicoid binding sites within those enhancers, as well as the nucleosome positioning within the target enhancers. And this story was published as of last week. Okay, so I'd like to thank everyone in the Blythe Lab, Neil Mangan, who is in part supported by NITMB, as well as my thesis committee, including Rich, and then my funding sources. And thank you for listening.
Questions?
Thanks for a great talk. Some origins of replication fire early and some fire late. So I was wondering whether you take that into consideration when doing the modeling, or whether you assume that all origins are firing in a single distribution.
The measurements by Blumenthal suggest that origins fire synchronously, or at least very close to synchronously. So it is still an assumption because I haven't measured it live, but yeah.
Yeah, I was just mentioning that because there are now live cell imaging measurements, at least in mammalian cells and reconstituting systems. And it seems that the origins of replication tend to fire in two subpopulations. Yeah, one fires very early, and then we have another population that fires very late. And there are some different characteristics of each of those populations. So.
Yeah. So for the Drosophila embryo, the cell cycles are so short. So in the case of my measurements, it's just a 20-minute time period. So there's not really time to have the late replicating domain. So it is unique in this scenario that you can just have simultaneous firing of the origins.
Thank you very much.